Students, a warm good morning to all of you. Welcome to the second session of the complement system. Previously, we had an introduction on what are complements, and we know that they are proteins, usually cell-bound proteins as well as soluble proteins, which have been involved in both the innate as well as the adaptive immunity. These proteins, called the complements, they are produced by the hepatocytes, monocytes, as well as the epithelial cells of gastrointestinal and the genitourinary tract, and they constitute about 5% of the weight of the serum globulin. Now, all these complements, they are being found as zymogens or proenzymes, which are usually inactive at, at the beginning, but when they are being lysed by proteins, the inhibitory fragment is being removed and the active site of these proteins will be getting exposed. And once the active site of the protein is getting exposed, they play a reaction which starts in an enzyme cascade. And what enzyme or what function does these complements do? The main function is that they trigger the immune response in a faster rate. Just like normal enzymes acts as catalyst for the conversion of a substrate to a product. The same way, the presence of a complement or an activated complement during an immune response triggers the immune system. And what all functions do the complements carry out? The first one is the lysis of the cells, bacteria, as well as viruses. And this constitutes a major effector of the humoral branch of the immune system. And secondly, it can also carry out a process called the opsonization, which promotes the phagocytosis of the particulate antigen. Thirdly, we can see that during the process of inflammation, you can see the complement receptors or that is onto the complement receptors of the immune system. The complement gets bind and that would trigger the inflammation process. And you can also visualize a process of immune clearance once antigen antibody complexes are being formed. These are removed from the circulation and they will be deposited into the spleen and in the liver with the further decomposition with the aid of the complement action. So these are the four different functions which I mentioned right now: the lysis, the opsonization, the activation of the inflammatory response, and the clearance of the antigen antibody complexes. And in all these functions, you can see that some proteins do take part and they are called the complements. And I hope you remember that we had previously mentioned that it was Jules Bordet who found out the complement system. And firstly, it was being produced when the action was being observed, when antigen antibody complexes are being formed. What happens to complete the or to trigger the antigen antibody complex mediated lysis, the complements were being found. And in first case, the complements were being described as proteins which would aid in the clearance of the immune complex. But that was not the only role of a complement, as we know that they also do play various other functions within a body. Now, these complements get activated by three different pathways. One is a classical pathway. This happens when a normal immune response has to be triggered when an antigen antibody complex is being formed. And there is another alternative pathway when cell surface constituents that are foreign to the host are being formed and it is not being found to be antibody independent. Now, alternative pathway, it is mainly found mainly when different types of proteins, like for example, it is an activator, then a polysaccharide from the yeast wall, that is cymosin, and then if you want, sometimes you have a cobra venom factor or a nephritic factor Okay, when all these factors comes, that is when your immune response have to be triggered in a very fast fashion, without waiting for long. The alternative pathway is being triggered so that we do not wait for the antibodies to be produced, but before that itself the complements act, and the alternative pathway gets activated. Now, why it produces a faster action, you will understand when you view the steps of it. Then finally, we have another pathway called the lectin pathway. The lectin pathway is another pathway activated when microorganisms who have mannose residues on their glycoproteins, or glycoproteins on their surface do enter into the body. And in such cases, what happens? Certain mannose binding lectins will be bound to the surface, and they will activate the lectin pathway. And this pathway is also being found to be antibody independent. Now, whatever be the pathway, I told you there are three different pathways, conditions, or the three different pathways by which the complements will be activated. Whatever be the pathway they get activated, the main important function or the important component is the C3 convertase formation. And once the C3 convertase is being formed, the different effector functions will be carried out, like anaphylaxis. Then a membrane attacking complex will be formed in some cases, and some cases like in inflammation we have the chemotaxis which is being carried out. And once a membrane attack complex is being formed, you can either have functional disruption or the death of the cells. So let's move on to the different pathways. Now, the first pathway is called the classical pathway. Now, I told you the classical pathway is the first pathway which we are describing, and the classical pathway is mainly being initiated when an antigen and an antibody complex is being formed. So, when a soluble antigen is being bound with an antibody, or when a bacteria is being bound with an antibody, what happens is some conformational changes will happen in the Fc portion of that particular immunoglobulin. You know, immunoglobulin is a Y-shaped structure. Okay, and on the Fc portion, the immunoglobulin will bind at the what? If you take the Y-shaped structure, the V portion or the ends of the V are the ones which interact with the antigens. The remaining portion, the stalk-like portion, is called the Fc fragment. So once an immunoglobulin gets bound to an antigen, the Fc portion will undergo some conformational changes. Okay, and it will expose the binding site on the CH2 domain of the Fc portion for the C1 component of the complement system. Do remember, the complement is not a single protein. Multiple proteins which are being found in the inactive form will get converted into the active form when the complements are being getting activated. So once, as I mentioned here, once an antigen antibody complex has been formed, the Fc, it's already bound to the antigen, and the Fc portion of that immunoglobulin is undergoing some changes. And onto that, what would happen? The first component of the complement pathway, the C1 component, will get bound. And C1 itself, it has three different components: the C1q, C1r, and as well as C1s. And do remember, in the first time or before the activation, these will be found in an inactive form, and they have to get activated. So what will happen is the activation will happen only when C1q is attached to the immunoglobulins by at least two of its binding sites. Okay, and one of the IgM or two molecules of IgG can initiate the process. So once C1q is being bound by two sides, see, you can see it has got two heads. Okay, so when either one IgM or two IgGs are getting bound to this by its Fc portion, okay, if it gets bound to the C1q side, okay, the C1q gets activated. And then onto this, what would happen? When calcium binding is also been happen, when onto the C1q, when calcium also gets bound to it, you can see that C1r and C1s also will come and bind onto it, and thus it gets activated. So at the beginning, you have the antigen antibody complex. Onto that, the C1q comes and binds, and calcium binds, and you have the C1 and C1r and the C1s. These are all proteins which get activated, and that would trigger the complement activation pathway, the classical. So you have C1q binds to the antigen bound antibody. Then binding of C1q to the Fc induces conformational changes in C1r, and the C1r gets converted into a serine protease enzyme called the C1r dash, which cleaves C1s to a similar active enzyme, and these three components, the active forms, will get. So this is a, you just see, this is a antibody, and it is bound to a bacteria, and onto the Fc portion of that, what is it? C1q is coming in binding, and onto C1q you have the activated C1r2 and S2 dash activated components bound. So this is the formation of the first complex. The classical pathway gets activated. Now, this is what we are at this stage now. Don't look into the other pathways, just look at the classical pathway. So we have the antibody here. Onto the antibody, the C1q is bound, the R is bound, as well as the S is being bound. Once the C1qRS, the activated forms of this are being present, they will go and act on the another component called the C4. C4 is also a complement component. And C4, what happens, gets split into two components, C4b and C4a. And in this, as I mentioned earlier, the complements always will be found in an inactive form. And when we split it with some enzyme, it will be converted into an active form. And only the active part of it will enter into the complement pathway. The remaining will move out. Okay. So in this, C4b is the one which is being needed for the complement pathway. So into the C1qRS, the antigen bound C1qRS, the C4b also gets bound. Okay. And there's another component here called the C4a, which has been formed. That will serve as a what? It's a pink color. See, look what is pink. It is a anaphylatoxin. So C4a will move out as an anaphylatoxin, but the antigen antibody complex, that is with QRS, onto that QRS, the C4b also gets bound. After that, the next component, the C2, that is the next complement. The C2 gets cleaved, and that also form two components called the what? C2a and the C2b will be formed, and C2a will enter into the pathway, and C2b is in fact a kinase that has not been depicted, but C2b, okay, it acts as a kinin and it moves out, and C2a enters into this complex. So do visualize like this. You have a bacteria which is bound with an antibody, then you have the C1qRS2 bound, and onto this complex, further what happens, the activated C4b comes and binds, then after that you have the C2a bound. So the C4b and 2a, onto that itself, these are all bound to the antigen antibody complex which has been formed over here. Now you have C4 4b and 2a, and that will further activate another protein called the C3 convertase, C3, okay, C3, and that will, C3 will get split into what? C3a and C3b. Do remember, just remember, after 1, 4 is coming, after 4, 2 is coming, then comes 3, okay, and C4b, 2a and 3b is coming. And once this is being formed, it further splits the C5 component. The C5 component is a what? It gets split into C5A and C5B. And 5A as well as 3A, you see these are going as anaphylatoxins. So you have C5B onto this 4B, 3B, C5 will come, 5B will enter. Then further on C6, C7, C8 and C9 components. These all join together and result in the formation of a membrane attacking complex. So we started with a classical pathway like this. You have the antigen antibody complex, and onto that the C1q binds, then C1r and S binds, and once they are being activated, they will split the C4 component and the C4b component gets bound to it. Then further the C2 gets split and it forms 2A, 2B, and in that 2A enters into the binds to the antigen antibody complex. Further, that acts on to the C3 and C3b gets bound to this complex. Further, C5 gets split and it gets C5b component also gets bound. Then component C6, C7, C8 and C9, these are all proteins, okay? They all get bound together and they form a membrane attacking complex so that they can lyse the target cell. So it's just like you have a bacteria and onto that an antibody is being formed, and in order to lyse it very effectively, multiple proteins are being coming attaching onto it in a sequential fashion so that the membrane attacking complex, that is different enzymes together, act on the membrane of this bacteria and try to lyse it. And the classical pathway is mainly being formed when what? A antigen antibody complex is being formed. Now let's move to the alternative pathway. In the alternative pathway, I told you it is a pathway, it is a shortened pathway which has to be act at the time of emergency. Okay. Now, for example, a cobra venom is coming, or if you have a yeast infection with the help of which contains like yeast cell wall will contain a protein called the cymosan. Okay, when all these things are being present, the immune response has to be triggered out very fast, or the membrane attacking complex should be formed very fast. So what does the cell do is it is not going to wait for what? C1q, C4, then C2, that is C1, C4 and C2. Instead, when you need fast action, the cell will get triggered by C3 itself. Okay, and that is what's happened. So, but instead of that, certain other factors will also act. So, C3 is being formed in the, it will be C3 is being formed within the body, and it will be formed in the inactive form. So, once an activator is being formed, what is the activator for the alternative pathway? It could be anything. It could be now, for example, a cobra venom factor or a nephritic factor or even a yeast cell wall polysaccharide. Cymosin is a polysaccharide, okay, not a protein. I made a mistake earlier. So, once that gets bound to it, the C3 gets converted into C3b and C3a. Okay, and onto this activated C3b, the factor B, another component that gets bound to it. Okay, this is called the factor B. Okay, and in the factor B, like you have the what? A BB component is being formed, is there. So you have the C3b and the factor BB that also gets bound to it, and also a factor D also comes and act to it, and it is the factor D which splits the factor B into factor BB. Okay, and what happens is, and BA and BB, factor BA and BB, BA is not been shown here. So you have the C3 fragment which gets activated by the cobra venom, nephritic factor, or the cymosin and converted into C3b and C3a. In that, C3b will come, okay, and onto that the factor B will get attached. Now, how factor B is being formed? Factor B is being split by factor D, okay, and that will get converted into BB, and you get the C3b, capital letter B, small letter b. These components will be formed, and after that this combines with the what? The C3. The C3 component is already there, okay. The C3bb1 further combines with again C3b components, and then it comes up what? C3b, bb, and the 3b components that is also been bound, and further what? A cascade fashion is also being formed within over, and the membrane, once this has been activated, it will convert the C5 into C5b, and then the 5, 6, 7 complexes will be bound, and then you have the cascade fashion is being triggered. So here you can see that without the C1, C4 and the C2, the C3 gets activated and the pathway continues over here. So in the case of alternative pathway, it acts at a faster pace, avoiding some of the proteins, and the immune response is being triggered at a faster rate. And let's see, in addition to the cobra venom and all that, there are also some other factors which will initiate the alternative complement pathway. Now, for example, some strains of gram-negative bacteria, the lipopolysaccharides or some gram-negative bacteria, many gram-positive bacteria, the teichoic acids from the gram-positive cell wall, the fungal and the yeast cell wall components called zymosins, as we had mentioned, the polysaccharides, some viruses and virus-infected cells or tumor cells, as well as the parasites, and even non-pathogens. Now, for example, you have like human immunoglobulins IgG, IgA, and IgE, which have been found in complexes, or rabbit and guinea pig uh, IgG in complexes, and as I told you, cobra venom factor, or the anionic polymers like dextran sulfate, or pure carbohydrates like agarose, inulin. All these, when they are being present, what happens? The alternative complement pathway will be activated. And let me just show you the alternative pathway once more. We have the C3 is converted into C3b and C3a, and further onto that the factor B and the factor D comes and act, and further you get this activated C3bb and capital letter B, small letter b. Onto that, what happens? For the stabilization of that, propodin, a component called the propodin, also gets bound to it, and that will further what? The C3 component will be further been broken, and then 3b, that is also been bound over here, and okay, and further on to this, the C5 and the remaining components bind, and it forms a membrane attacking complex, so that the what? The alternative pathway is being formed. Now, next pathway which we have to talk about is a lectin pathway. Now, when does a lectin pathway happens? Lectin is a protein that will bind to carbohydrate. And as I told you, certain on the surface of certain microbes, they do have some mannoses. Mannose residues will be found on the surfaces, and onto these mannoses, which have been present on the microbial surfaces, the mannose binding lectins or the proteins, which are these MBLs. Okay, these are in fact acute phase proteins, and they will bind onto these mannose residues which are being found on the surface of certain microorganisms. And these mannose binding lectins, they are in fact structurally similar to the C1q. In the classical pathway, we had seen the C1q. So MBL is similar to that of the C1q. Okay, and Just like onto C1q, C1r, and C1s's binding, associated with this MBL, certain other proteins called the MASP1 and MASP2 can also get bound. And they are structurally similar to C1r and C1s. And what is this MASP1 and MASP2? They are MBL associated serine proteases. So once they are getting bound to it, okay, so you have a microbial surface on which has mannose. Onto that MBL comes and binds, and after that MESP1 and MESP2 will get bound to it, and you can see that they will function just like the C1QRS complex. Okay, and lectin pathway, just like the alternative pathway, it is an antibody independent pathway, and it is an immune defense mechanism comparable to the alternative pathway, but the elements of the classical pathway except the C1 proteins are being utilized over here. So now let's see what are the different components which are being formed within it. I'm just taking the one which you have seen earlier, the slide which you have seen previously itself. So you have the microbe which is having a mannose residue. Onto that MBL gets bound, or the mannose binding lectin, and also another component called the ficolin also comes and binds, and associated with that what the MASP1 and MASP2 comes and binds, and by the action of these serine proteases you can see C3 is converted into C3b, and at the same time C4 is also being converted into C4b and C4a, and these active components of C3b and C4b also combines together. 4b and 3b is there, and under the influence of this also you have the C2 component which is also being missing, and that will also bound, and the C4b, 2b and 3b complex will be found. Into this the C5 also gets split, and they will form the what? C5b, then further on C6, C7, C8 and C9 is also being formed. So here, instead of the C1 complex, what the MBL and the MASP1 and 2 gets bound to it. And with like that, the lectin pathway will get activated. So the difference is that instead of C1, we have the what? The MBL as well as the MASP1 and 2 which is being formed. You can see another representation of the same thing which we are having here. You can take the one which you have I think this is a bit you have the mannose binding lectins over here, and that has been associated with MASP1 and MASP2, and further it is activating various other components like the C4, C2 as well as the C3, and you are getting the what? C4b, 2a, 3b complex over here. Onto that the 5, 6, 7, 8, 9 comes, and then it becomes a membrane attacking complex as we had mentioned in the other thing. So I guess right now you are clear with the different pathways which have been present within the what? In the complement activation, and this is a picture of a complement a membrane attacking complex which has been formed on the cell surface. Okay, in vitro you can see the lysis of the cell is being caused about with the help of the complements. This is another picture where you can see the alive cell looks like this, the killed cell looks like this. So with the help of the complements, the lysis and killing of the cell will happen. And of course the regulation of the complement system also is being present because some of them, I told you, only in some cases what is it, you need to activate the what? The complement pathway, and once the antigen antibody complexes are not there, the complements are not to be needed. So in such cases inhibition of the complement pathway will also be carried out, and when the complements are needed they will be activated and they will be produced. So with this we come to an end of the complement system. Thank you for now.